pests with all the ensuing damages. But damage caused by pests goes beyond direct economic impact. It is also one of the major factors of generating biodiversity loss. The introduction of new species leads to crowding out or destruction of native species. For Xylella fastidiosa, the wide negative environmental impacts have already been well demonstrated, and also the impacts on rural communities and our cultural heritage. I find it is also important to place plant health and Xylella into the bigger picture of the current policy agenda of the European Union. Within the European Green Deal, both the biodiversity strategy and the farm-to-fork strategy aim at transforming the agri-food system towards a sustainable operating model. It is an ambitious plan, spanning from primary production to healthy choices for and by consumers. We all know that this is essential for the future of our planet, to not only provide safe food, because if it's not safe, it's not food, but to consume food within balanced diets and to produce food in a sustainable way. The future in 2050 will see us having to provide food for approximately 10 billion people. This seems doable, but three major challenges have to be overcome. To increase crop-based calories production by roughly 55%, not to increase land use for agriculture while restoring biodiversity, and in parallel, to reduce greenhouse gas emissions from the agri-food system by roughly 2/3 compared to the 2010 levels. To achieve this, a profound transformation of the agri-food system is needed, spanning from increasing supply, coined as sustainable intensification, to reducing demand by tackling food loss, food waste, and by profoundly changing dietary habits. In my view, this is one of the biggest challenges ahead of all of us, for science, producers, policymakers, civil society organizations, and consumers alike. We at EFSA will continue to play our role in providing scientific advice on some of these important issues. We will be able to do that only by working together with national authorities, universities, and research centers. The key word for all that is collaboration. And I have to share with you, not many things strike me more kind of strange after 30 years in this business than the still underutilized opportunities of cooperation. There's nothing more powerful and rewarding than to deepen interactions, from getting connected to one another via cooperating on projects to co-creating the future together. I mean, intellectually seen, this is a no-brainer. Everybody would agree on it. But in the real working world, it's still not done enough. Having this in mind, it is truly satisfying to look at today's conference. Here, we do pool our resources, exchange our research results, and tackle problems such as Xylella together. This approach greatly increases the chances to solve the issues we are facing. This online event, together with the XF Actors final meeting preceding it this week, is convening around 900 registered participants from 64 different countries. Approximately 80% are from Europe, from 24 EU countries and from 9 outside EU countries. 9% of participants are from the Americas, from 10 countries, 5% from Africa, 7 countries, and many colleagues from Asia, 12 countries, and Oceania, 2 countries. Participants are affiliated with universities and research centers, more than 50%, the private sector, NGOs, farmers and nurserymen associations, national plant health organizations, international organizations, risk managers, and risk assessors. You are all very much welcome. This collaborative third European Xylella conference aims at building knowledge, protecting plant health. Between today and tomorrow, there will be 39 talks on the stage, including 12 talks by young researchers, and 100 posters will be presented. The conference will end with a discussion with stakeholders on their research needs and priorities. We really look forward to an interesting and lively debate that will help us all to better understand the available solutions and the future research needs and priorities to fight Xylella. I wish you a fruitful conference, and not only in the sense of fruits from healthy plants, but even more so in the sense of fruitful human encounters. In my opinion, it is these encounters that build the trust for a deep cooperation. Thank you very much and all the best.
Thank you very much, Bernard, for the opening to this conference. We will now proceed with a brief introduction to the content of the conference, and then at 2:30 we will start the first session, which is about genetics and biology. Okay, so this is the third European conference on Xylella fastidiosa, and it follows from the first event, which was organized by EFSA European project and precede the conference for 3 days. It's co-organized together with several organizations, as presented by Bernard. We all have attended in the last 3 days the three full afternoon of the EFSA final meeting, which were concluded yesterday at 7 o'clock. Now ahead of us is the 2 sessions today on advance of pathogen biology and genetics, and the session on vector biology and vector pathogen transmission. Tomorrow we'll have 3 sessions. Session 3 from epidemiological modeling to surveillance and early detection. Session 4 toward the sustainable management strategy for Xylella fastidiosa. Session 5 will be a discussion with stakeholders on their needs and the priority for research on Xylella fastidiosa. And then we'll finish at 6 o'clock till 6:15 tomorrow with a conclusion. The next session today is about advanced pathogen biology, ecology, and genetics. It starts at 2:30. Will be chaired by Ralph Kebnick from the Plant Health Institute of Montpellier and the Institute for Research and Development from France. Ralph Kebnick is also leading the Euroxant cost action. And the session will be chaired together by Massimiliano Morelli from the Institute for Sustainable Plant Protection from the National Research Council of Italy. Me and Alice Del Bianco will moderate the session through dispatching the online question to the chairs. And we will have a total of 9 speakers, including 3 young researchers and 3 short question and answer sessions. Then we'll have a short coffee break at 5:05, 15 minutes, during which on the screen you will be able to watch interviews with young researchers, which are also published on YouTube. And then at 5:20, we start the session 2 to understand the vector biology and the pathogen transmission. Chaired by Astrid Cruot from INRAE, France, and Saskia Huguenot from John Innes Centre, UK, who is also leading the Bridget Consortium for Research in UK against Xylella fastidiosa. The session will be moderated by Evelina Cvencic and Ciro Gardi from EFSA, and we will have 8 speakers, 3 young researchers, and 3 short question and answer sessions. I would like to remind that we will continue having a poster session that starts on Monday online. You can find all the poster if you look for the hashtag Xylella21 poster in Twitter, and you can interact with the author by replying to the original tweets by plants at EFSA. And by commenting and replying to poster, you will contribute to the poster competition. As you can see here, the Twitter that report always the DOI of each poster in Zenodo. You can find the DOI of all the poster in the program or the conference. And how it works with the social media poster competition, we will reward the author the most interesting or best presented work by looking at Twitter analytics. So we'll calculate using the social interaction value, the number of likes, retweets times 2, and number of replies that will value 3 times. And the winner will be announced tomorrow on the morning at 12 o'clock by Sara Tramontini. The winner will be interviewed on the web, and the interview will publish on EFSA YouTube channel after the conference. I also would like to remind you how to interact during the conference. This is an online conference. Unfortunately, we cannot have open question to the speaker. The question to the speaker will be put online to the Q&A box that you all participants should find on their screen. And you have to type the question and then select all panelists. The question will be sent to the panelists. And during the conference, the moderator will select the question sent to the chair that will ask them orally to the speaker during the Q&A session. And if we don't manage to reply all the questions during the event, this will be done after the conference in the proceeding, also grouping similar questions. In general, we'll try to reply all questions, but as the event is about research, the question outside this topic may not be replied. So the last technical aspect, normally when you access the link, you should not be asked for password, but if you ask for password, this is a complicated one, which was just changed today, respect to the simple one we have yesterday. So please pay attention to this. You also receive by email. And if you have any problem, you can write to Xylella conference. So thank you very much, everybody, and enjoy today's session. I think we're a bit ahead of time, but I will pass the floor to the chair of the first session to introduce the content of the session.
Yeah, thanks a lot, Giuseppe, for these opening words and given explanations. I would like to share my screen. Is this possible?
Ralph, can you also put your video?
Ah, sorry.
So yes, we see your,
I can share my screen.
Your face, yeah.
Okay. So I wonder, maybe we should still wait a few minutes before we really start with the first session. But I may introduce another event. So hello, everybody. I am Ralf Köpnick from the Plant Health Institute of Montpellier in France. Welcome to this first session of the third European Conference on Xylella fastidiosa. Before opening this session, which will address advances on pathogen biology, ecology, and genetics, I would like to inform you about another upcoming conference on Xylella and its relative Xanthomonas. This conference is taking place by the end of June and is organized by the Euroxant Cost Action, which I am chairing. If you feel interested in joining this conference, please follow the links on our website. More information about the conference can be found under Events, and registration is possible under Forms. Please be aware that the deadline for registration is on May 9. Now let's come to the first session of today's conference, which is under the sponsorship of the European Food Safety Authority. The first session will be chaired by me and my colleague, Massimiliano Morelli, from the Institute for Sustainable Plant Protection in Bari in Italy. We will start with 3 presentations in a row, followed by a discussion where we will discuss all 3 presentations at once. You can submit your questions in the chat on your screen, which will then be pre-collected by the moderators and passed to the speakers by Massimiliano and me. It's our pleasure to welcome our first speaker, Aina Barro from the University of Girona in Spain, who will talk about Spanish isolates of Xylella from almond trees. The floor is yours, Aina.
Thank you very much. Okay, so hi everyone. Thank you very much for giving us the opportunity to present our last work on Xylella fastidiosa in this 3rd European conference dedicated to this important pathogen. Today, I'm going to present some results related with Xylella fastidiosa isolates into almond cultivars. And maybe the first question is, why in almonds? So the choice is basically because almond leaf scourge was first detected in Mallorca in 2017, which was associated with the isolates from the sequence type 1, 7, and 81. Also, Alicante is the Spanish region affected by Xylella fastidiosa, but in this case, these isolates are from the sequence type 6. Almond leaf scourge is also present in Apulia, where isolates from the sequence type 33 have been detected also in almond. So almond leaf scourge, as you can see, is a very important disease now affecting almond crops in Spain. Although there are already studies about the pathogenicity of Xylella fastidiosa strains in almond plants, but due to the emergence of new genotypes in Europe and the observed variability of this pathogen, we consider it important to study the Spanish isolates affecting this crop. In order to achieve our aim, we designed an experimental approach based on the artificial inoculation of 1-year almond plants. Although it's worth it to consider that this experimental approach does not represent the agronomic conditions in most of the current almond plantations, this system is comparable to the situation in nurseries and super-intensive orchards. In addition to the 2 almond isolates responsible for the outbreaks already mentioned, the one from Mallorca and the one from Alicante, we consider it also important to add in the study isolates from cherry trees from the sequence type 1, and also isolates from grapevine from the sequence type 1 again. All Xylella fastidiosa strains were inoculated using a high-precision microinjector provided with a Hamilton shrink and with a needle, with a thin needle with a pebble tip, as you can see in this photograph here. This system allowed us the accurate application of 3 inoculations of 10 microliters of inoculum in a reproducible way in each plant, as you can see in the scheme, being able to precisely control the dose of inoculum. Here you can see a detail of the process of inoculation. The needle line was introduced in the plant stem in order to directly inoculate the vascular system. as our aim was to mimic as much as possible the feeding behavior of the insect vectors. Sampling of the infected plants was performed at different time points, and population levels of Xylella fastidiosa were quantified in order to determine the movement and the multiplication of Xylella fastidiosa from the inoculation points to the other plant parts. First, the first step of the processing was removing the bark from the stem. And then from this point, we analyzed different plant matrices. In one hand, we quantified the viable fraction of planktonic cells in sap, and the procedure followed was the one here, followed with green arrows. And in the other hand, we quantified the total fraction of cells in the xylem tissue, following the process that we can see here in red. In both cases, what we obtained was DNA from the plant material, which was analyzed by qPCR in order to detect Xylella fastidiosa. In order to differentiate between viable cells and non-viable cells or dead, we used viability qPCR, which briefly is a technique that is based on the treatment of the sample with acid nucleic binding dyes prior to the DNA extraction and quantification by qPCR. In our case, we used the reagent PMA-6, which is a dye that binds to the free DNA and the DNA from dead cells with damaged membranes, avoiding its subsequent amplification by qPCR. We performed standard curves in order to be sure that sap content was not interfering with the reagent. Okay, so now I'm going to pass through the results we obtained during this study. Here in the first figure, we present population levels of Xylella fastidiosa in the 3 plant parts analyzed and over a period of 6 months. Also, we present the disease severity associated with these plants. The dynamics of the almond isolate from Mallorca in almond plants of the cultivar Abijur showed 2 clear different phases. In the first phase, we found an exponential growth either in sap and xylem tissue with doubling times around 2 days. And in this case, the disease severity during this phase was very low. Only a few leaves were showing marginal necrosis. In a second phase, what we found was that Xylella fastidiosa cells remained stable in the stem, as you can see here with the dashed line, while Xylella fastidiosa cells decreased in sap. The values are the ones with the solid line. During this phase, regarding symptoms, what we observed, it was an increase in the disease severity that had a maximum at 120 days post-inoculation. After determining this time course of population levels and symptoms, what we did was studying the colonization behavior of Xylella fastidiosa isolates on the 3 almond cultivars and in the 3 plant parts analyzed. Here, what we observed was a different colonization behavior, being the isolate from almond trees in Mallorca, the one showing the highest population levels, followed by the isolate from cherry trees. The isolate from Alicante showed the lowest bacterial load, as you can see here. And regarding the grapevine isolate, it was able to colonize almond. And in the case of cultivar Penta and Xylella, this isolate reported the similar population levels that were observed with the other strains of the subspecies fastidiosa. Here we present the disease severity related with these plants that we have already talked about. What we observe here is a different susceptibility of to the four Xylella fastidiosa isolates, being Penta the most susceptible one, showing a higher rate of symptoms, while Abijur was the least susceptible. Regarding strains, the grapevine isolate showed the highest rate of symptoms, while the isolate from Alicante showed very low symptoms in Penta and Xylella and none in Abijur. Also, in these plants, we considered interesting to analyze the capacity of spread of these different isolates to the new growing shoots after pruning. Here we present the population levels of each strain in each almond cultivar. First of all, what we can see is that Xylella fastidiosa is able to move from contaminated plant parts to new growing shoots. And in general, and in agreement with the results obtained in the first sampling, almond isolate from Mallorca reached the highest population levels, while the isolate from Alicante showed the lowest population levels. Also, in agreement with the other results, Penta was the cultivar showing the highest rate of spread to the new shoots. Now, and in order to compare the two almond isolates, we performed those effect relationships, which are represented in this figure. Here we represent population levels against inoculation doses in the three parts analyzed and for the two almond isolates. For each strain, what we see is that the relationship between inoculation dose and population levels follows similar patterns of saturation in each plant part analyzed. And also for both strains, high inoculation doses reported high population levels. When comparing the two strains, what we saw is different strain performance. While the isolate from Mallorca reported higher population doses, it was also seen that this isolate had median effective dose high. On the contrary, the isolate from Alicante attained lower population levels, but its median effective dose was also estimated low, indicating that this strain needs low doses in order to achieve the 50% of its maximum population level. Regarding minimal effective doses, although they were different between strains, they were both below 3,000 colony forming units per plant, indicating that vectors could easily transmit the disease, as concentrations ranging from 30 to 10 to the 5 colony forming units have been found in insect heads. Nevertheless, it's important to say that transmission is not only affected by those, but also by plant host, insect species, and pathogen strain. Okay, so now to summarize a little bit the importance of our study, we consider that it raises implications in the epidemiology of Xylella fastidiosa directly in almond crops, but also in other plant hosts. Differences in susceptibility to Xylella fastidiosa infections were found among almond cultivars, as well as differential aggressiveness among Spanish strains isolated from different plant hosts. Additionally, almost all Xylella fastidiosa strains had the capacity to spread from pruned infected tissues to new growing shoots. In the case of the 2 almond isolates studied, low Xylella fastidiosa doses were required for infection. And regarding control, we consider that these results can be useful information for the optimization of disease management guidelines. Okay, thank you very much for your attention, and many thanks to my colleagues from the University of Girona and to all the projects and grants that have funded this research. And just to say that if you want to know more, these results are part of a paper that has been recently published in the journal Phytopathology. So I invite you to take a look at it. Thank you.
Thank you, Aina, for your interesting talk, and good afternoon to everybody. I'm Massimiliano Morelli, and as Ralph already mentioned, I will co-chair this session that I guess will be of great interest for all our audience. So now let me to introduce the next speaker, who is Michael OLeary from US Department of Agriculture and Agricultural Research Service in the US. Michael will present his most recent findings on type 1 restriction modification systems and methylation patterns in Xylella fastidiosa. Please, Michael, go ahead. The floor is yours.
All right, let me just get my screen set up here. Okay, did that show up?
Perfect.
Screen showing up for everyone, yes?
Yep.
Okay, great. So I certainly want to thank the organizers for giving me the opportunity to talk about the kind of work that I've been doing. So what I'm going to do today is talk a little bit about restriction modification systems in terms of what they're doing and what roles they can potentially be playing in bacteria. Sure, I can start my webcam. Let's see. Actually, I'm not sure how to do that once the session is going. Sorry, I'm not sure how to start that when I'm sharing my screen. So anyway, so I'm going to also talk a little bit about, or mostly about type 1 restriction modification systems in Xylella, where there's a fair amount of variation between a particular component called the specificity subunit. And then I'm going to talk about the work that I've been doing trying to identify some genomic methylation patterns across different Xylella strains. So fundamentally, restriction modification systems have 2 components. These are a restriction component and a methyltransferase component. These operate on the same target site. Commonly, when we're talking about the most common types that we encounter, type 2 restriction modification systems, this is like a 6 base pair, often a palindromic site. The methyltransferase modifies this site by adding a methyl group, and the restriction endonuclease will cut that site if it doesn't have a methyl group. Functionally, what this means is it provides some defense against foreign DNA elements because within a cell, all of the restriction modification systems are going to modify all the DNA with a particular pattern, and foreign DNA is not likely to match that pattern and will be vulnerable to being cut by the restriction enzyme. And this can provide defense against phages, or mobile elements, but also contributes to genetic isolation through that same method. Strains that don't have the same restriction modification systems presumably have a more difficult time exchanging genetic material. This is thought to contribute to maintaining species identity in bacteria. But methylation itself, which is mostly what I'm going to focus on here, can have some other roles. One of those is transcriptional regulation. This is really common in eukaryotes. We're finding out more about this in bacteria as time goes on. One way this can work in bacterial pathogens, and this is mostly in animal systems, is through phase variation, which is reversible on-off switching of certain genes. This can lead to the formation of subpopulations within a clonal lineage, even on the same culture plate. And altering methyltransferase activity does affect pathogenicity in some bacteria. These are mostly animal pathogens. This is where most of the work has been done, although I've seen a couple of papers over the last few years talking about this in Xanthomonas as well. So the original genome papers with Xylella strains noted that they're enriched in restriction modification systems. And this has remained true as more Xylella genomes have been released. Typically, there's around 3 times as many restriction modification systems as you would expect based on the genome size. So there's some variation here between subspecies and clades in terms of which particular ones are present or what their closest homolog is out of NEB's restriction enzyme database, enzymes with known activity. But what stands out to me here are all of these purple columns. These are type 1 restriction modification systems. So depending on the strain, there's 3 to 4 of these. 2 of them are present in every strain. These are so they're conserved and they're adjacent to each other in the genome. They're separated by another probable endonuclease. The third one is present only in multiplex and Pauca strains, as far as I can tell. And there's a 4th one that seems like it's present in pretty much every strain, but it's very often inactivated through multiple mutations. So I'm not sure how important this one is or what the role of that one is. All right, so type 1 restriction modification systems are fairly complicated. They add a 3rd subunit to this restriction and modification system. This is a specificity subunit. So without this, the complex doesn't function. You could consider it the linchpin subunit here. So there's 2 complexes that can be formed. This is a subunit, specificity subunit along with methyltransferase, and this will methylate DNA at hemimethylated sites. That means where one DNA strand has a modification and the other does not. And then a second complex can be formed that's all 3 subunits together, and this will cut unmethylated DNA. But it doesn't do it at the site that it recognizes. It will do it anywhere from 100 to thousands of base pairs away. The specificity subunit will guide the complex to a bipartite site. This is a short 3 to 4 base pair sequence with a non-specific spacer that can be any base, typically somewhere from 4 to 8 bases, followed by a second 3 to 4 base pair sequence. So the way that works is the specificity subunit has 2 target recognition domains, TRD1 and TRD2, and each one of these corresponds to half of that bipartite motif. So TRD1 in this hypothetical example recognizes this sequence GATG. There's this non-specific spacer. This is a core conserved region. The size of that relates to the size of the spacer. And then there's another 3 to 4 base pair sequence. Now, different TRD domains with different specificities have a drastically different sequence. If you align them at a DNA and often protein level, they look like they're completely unrelated sequence, although the core conserved region is highly conserved. And when you have a recombination event where these target recognition domains are exchanged, you end up with a novel allele that has a new specificity. Like in this example, designated the top TRDs A and A, the bottom B and B, and you can see there's this new allele BA. This is actually really common in these genes. You can do this experimentally in the lab and see this variation in bacterial populations as well. So on the right is a DNA alignment of one of these systems. This is XFA1. This is one of those conserved systems between two California sequence type 1 strains, M23 and Stag's Leap. So you can see that the methyltransferase subunit, HSDM, and the restriction subunit are conserved, but in this alignment, there's this gap that corresponds to part of the specificity subunit. And this is suggestive of TRD exchange, right? So the system, even though it's conserved, is probably recognizing different sites in these two relatively closely related strains. Okay, so there's a little bit of variation, but how much is there actually across Xylella as Xylella fastidiosa as a species? So what I did is I looked at the target recognition domains in each specificity subunit for each system, and I gave them a letter designation, and these are major groups that don't have homology to one another. The way to read this plot, it's a little busy. Each column, there we go. Figured out how to get my video on. Each column is one target recognition domain. So pairs of columns are going to actually represent the entire gene. But overall, what I want to show here is that there's a fair amount of variation between the subspecies of Xylella fastidiosa. That's where the majority of the variation is. And there's different levels of variation in the different systems. So some individual target recognition domains are shared between between strains, and definitely between strains and between subspecies. But intact alleles are very rarely shared. Now, if you consider the profile as a whole, they're typically limited to genetic clades, and this can be sequence types in some cases, and they're rarely shared between sequence types as well, although there are some cases of that. One thing that is interesting to me here is the sequence type 6 strains, which are a polyphyletic group, are differentiatable at these sites. So overall, this suggests that particular lineages are distinguishable by their specificity subunit profile, and that there may be some epigenetic differences between lineages. So how to look at that? So one way you can do that is with third-generation sequencing technologies. These, either PacBio or Nanopore, these can both detect DNA modifications. So I use Nanopore for that. I've been doing a lot of Nanopore sequencing. And the way this works is Nanopore recognizes records an electrical signal as DNA moves through a nanopore. And modified bases and unmodified bases have a different electrical signal. And you can compare genomic DNA versus amplified DNA from PCR, and you can compare that, and you'll get signal differences per site against an accurate reference genome, and can come up with modified motifs. So I looked at 12 Xylella strains. These are representatives from 3 subspecies, 7 sequence types, and 9 of the specificity subunit genotypes that I identified. Altogether, I was able to identify 15 unique bipartite motifs. This is this first box here. These are existing in 8 aggregate patterns. Now, what I thought was interesting here is that there were actually fewer motifs than systems. But these patterns do correlate with the differences in their HSDS profile. Some of the systems might not be functional. So, for example, sequence type 81 here, Fillmore, along with two of the sequence type 7 strains that I used, RH1 and LM10, have the same profile, but the same methylation pattern, but the difference in their profile might be at a system that's not actually expressed either under my experimental conditions or might be non-functional. I also identified type 2 and type 3 motifs. One of these was identified in a previous study as being a subspecies fastidiosa specific motif, although I did detect it in a Morris strain and a multiplex strain as well. In terms of those systems, there's definitely some differences, some bigger differences, more striking differences between subspecies, and a little bit less between strains. All right, so just in summary, there is a substantial variation among the specificity subunit genes in Xylella fastidiosa. And these specificity subunit allele profiles tend to correlate with genetic clades, and often, but not always, with sequence type. There's also variation in genomic methylation patterns at a subspecies and strain level. But overall, for both type 1 and also type 2 systems, I identified fewer modified motifs than methyltransferases. And this could be a limit of the approach that I used, or this could have to do with the experimental conditions, or sometimes these genes can be reversibly switched off. So something like that could be going on. There's some potential applications that immediately came to my mind for this. One is Xylella can be difficult to transform, and at least informing your construct design by the methylation patterns that are present in the genome of the strain you're working with might help. The results, at least for the type 2 systems, are fairly consistent with what's predicted in the restriction enzyme database. There's also possibilities of construct pretreatment. I know a CG methyltransferase is used somewhat often, as well as type 1 restriction modification system inhibitors, but other enzymes might be helpful. And it might also be possible to separate strains in a metagenome sample done with a 3rd generation sequencing technology by a methylation pattern. So, and just, I want to say at the end, I've got to acknowledge Jianqi Chen, who provided a lot of the strains that I used in this work, as well as some technicians who helped with various aspects of this project, Kumbo, Brandon, and Yadira. So thanks so much.
Thanks a lot, Michael, for this interesting guidance with the world of restriction systems and Xylella. We will have our next speaker, Nancy Walker, from the Faculty of Engineering and Physical Sciences at the University of Southampton in the UK, who will speak about model and image-based investigation of Xylella within host dynamics. The floor is yours, Nancy.
Okay, thank you very much, Ralph, for the introduction, and thank you everyone for this opportunity to present my work. I will start sharing my presentation. Okay, excellent. Okay, so good afternoon, everyone. My name is Nancy. I am from the University of Southampton in the UK, and I'm going to be giving a very brief overview of my work on Xylella fastidiosa within host dynamics. So as we all know, Xylella causes disease by forming biofilms, which act to restrict the flow of water in host xylem. However, the details regarding the exact mechanisms of this, and specifically how this differs in resistant hosts, is still not well characterized. So the main hypothesis of my work is that One such mechanism of resistance, specifically in olive cultivars, might be differences in xylem geometry. So in particular, we hypothesized that resistant cultivars would have larger diameter vessels. These would be able to conduct more water and would also be harder to block physically by biofilm formation. So to investigate these hypotheses, I'll be using both 3D X-ray computer tomography. This will enable visualization and measurements of differences in aspects of xylem geometry. Additionally, I will be developing a mathematical model of biofilm formation and development that will basically act to simulate the development of disease on these different xylem networks. So to start with the X-ray CT imaging, I have currently scanned 3 replicates from each of 4 different olive cultivars, 2 of which are susceptible and 2 of which are resistant to Xylella fastidiosa. To analyze these networks, I've developed a custom image processing workflow, which basically involves isolating the vessels using a grayscale threshold. And then from there, I can start making relevant measurements to look at different features that I'm interested in. These networks will also heavily aid my model parameterization and will also eventually form the spatial domain for more complex 3D simulations of the model. So initial measurements of the CT scans have focused on vessel diameters. We found that there was no statistically significant difference between the mean vessel diameters of the different cultivars that we looked at. However, when we looked in detail at the individual vessels, which is enabled by the detail provided by the CT imaging, we did see that there was a difference in the distribution of vessel diameters of the replicates that we looked at. And in fact, the distributions was the complete opposite of what we expected. We found that the susceptible cultivars actually had larger diameter vessels than the resistant cultivars. So on this basis, I want to investigate other morphological features that might be actually more indicative of actual resistance mechanisms. So onto the mathematical model. So I've developed this model on the pretense that the dominant component in determining the 3D structure of the biofilm, which is ultimately what is blocking the flow of water, is the presence of the EPS molecules. So on this assumption, I approximate the biofilm as a water polymer mixture, which is itself surrounded by water that's flowing in the xylem. So to come to my equations, I basically construct an energetic description of this polymer water system, and that results in 4 equations. The first 2 equations, as highlighted here, correspond with the organization of biofilm and the separation of the biofilm from the flow in the xylem. And the bottom 2 equations, they correspond with the actual movement of these 2 parts of the system, the biofilm and the xylem flow. So here I just show you some preliminary simulations of the model. So we have organization and growth, and we compare these with actual Xylella biofilms. So as you can see, the organization of biofilm is quite nicely already captured by just the polymer physics that's contained in the first 2 model equations that I showed you. However, the inclusion of growth still needs more accurate parameterization and further refinement. So just to conclude, the CT results of our first replicates showed unexpected result in regards to the vessel diameters, and as such, additional morphological features of xylem need to be investigated. Model simulations are able to nicely capture spatial organization of biofilm. However, more accurate inclusion of growth rates need to be added, and ultimately we want to apply this model to a CT-based geometry that we have collected from our CT imaging. So finally, I just want to say thank you again for listening, and just to acknowledge my funding from the Natural Environmental Research Council and supervision from the University of Southampton, UK Centre of Ecology and Hydrology, and Siena in Italy. So thank you very much.
Thank you, Nancy. We are perfectly on time, so we have now about 15 minutes to answer some of the questions that came from the attendees. So I'd like to call again our speakers on this virtual stage. And I will address the first question to Aina. This question came from the experience of Maria Saponari with olives. And Aina, Maria was asking and reporting that in our experience with olives, we found a strong correlation between root colonization and symptom expression. And we would like to know if you have checked if this may be also the case for almonds.
In our case, we haven't checked root colonization, so we cannot say if there is a correlation or not, but maybe it's something to take into account in future research, if you have seen this.
And also relative to this question, we would like also to know if have you tried in your experience the hybrid GF677, as it was found to be immune in our hands with the ST53 strain.
We tried it at the beginning, and we observed some kind of resistance compared to the other almond cultivars that we have already presented. So yeah, in our case, it also seems that it's more resistant. And we already checked the literature, and it's also reported this for this hybrid. So yeah.
Okay, thank you.
Okay, thank you. I have a question for Michael coming from Leonardo de la Fuente. Are methylation patterns influenced by the environment? Do you expect different patterns inside the plant or the insect versus lab media?
Right now there isn't any information on that in Xylella. In some other systems, you can see some changes based on media used and some environmental changes, I believe. It's going to depend on what transcription factors are turning them off and on, I think. So yeah, right now I'm not sure. I wouldn't be surprised if that's the case, though.
Okay. I have also a question that came for Nancy, and they are asking if there are, in your knowledge, other pathosystems where XCT-based geometries have proved useful for selection and individuation of resistant germplasm.
Yeah, I'm not too aware of any specific examples of this, but yeah, it's specifically XCT for this kind of research in plant pathogens. I haven't seen any similar studies so far.
Thanks, Nancy. I have another question to Aina. So you have observed differential aggressiveness between your strains quite significantly. Do you have any explanation coming maybe from genomic information why this could be the case? Do we have genome sequences for these strains?
No, we haven't done any genomic analysis, but we have thought that it could be interesting also because when comparing these two almond isolates, we have seen this different performance. So maybe it's something that we should do in future research also to compare both of them.
Thanks a lot.
Thank you.
I have also a question for Michael. Michael, as you mentioned, these type 1 restriction modification systems are, of course, also a signature of environmental presence of Xylella with other aspects. And I was wondering if you have any hypothesis on the fact that maybe even a plant host can somehow hijack these systems, and this could be maybe related to differential responses by the host.
So if I understand correctly, you're asking if the hosts can detect the activity of these systems?
Yeah, somehow interact with this. And
I mean, my sense from reading the literature is that these systems can sometimes influence gene expression. So if they are influencing gene expression, I think any degree of proteins or some change in expression could be detected perhaps. But the activity of the system itself, in terms of how it's modifying DNA, I doubt that. The system's it's not secreted or anything like that. It's a internal modification for the bacterial genome.
Okay, thank you.
I have another question to Michael and maybe to the community, even a very practical one, because you gave advice on kind of taking care when designing constructs. I wonder if people have used the OCR protein from the bacteriophage T7, which is an inhibitor of restriction enzymes, and if this is helpful in transforming Xylella.
So I think if I'm thinking of the same one, I think that's actually a commercialized type 1 restriction modification system inhibitor.
Yes.
Which there was a paper, I think around 10 years ago, it's quite a while ago, that showed that there's an increase in transformation efficiency when you use that. So part of that is what kicked off this work, so that observation. So
thanks a lot.
I have another question, maybe to Aina. And Aina, you showed us those effect relationship. And I was thinking if you could anyway predict the results of this analysis, and if this can be merged with the data collected by our colleagues working on vectors, and if this can help somehow to establish a kind of those effect in a correspondence to the action of the vectors according to the ability of the insect to carry Xylella cells, and they're likely to behave as a syringe in your case. If this can somehow help also study of vector and then epidemiological studies, of course.
I think that it's important to see these those response relationships, because then you can estimate this, the median effective dose, and then you can use these different values to use it in epidemiology, or for example, look which strain could be more prone to be spread for insect vectors if this value, for example, is lower than another strain. And from the first answer, if we can predict other strains, looking at the results we have obtained, I'm not sure if you can now predict, but maybe if you do some kind of genetic analysis or maybe from the same sequence type, it could be possible that strains can have a little bit the same pattern.
Okay, thanks.
Thanks.
It seems we are ready to start the second part of this session. And I'm pleased to invite to the floor Ofir Bahar from Agricultural Research Organization, Volcani Center, Israel. Dr. Bahar will update us with his current research that came out from the recent identification of Xylella fastidiosa in Israel. And I'm sure that this talk will be of great interest for all participants in these meetings. Please, Ofir, you can start sharing your screen.
Thank you very much, Massimiliano. Can you see my screen well?
Yes.
Yes.
Okay, so thanks again for the introduction and the invitation to speak here and to present our work. I'll be talking today about the distribution dynamics of Xylella fastidiosa within almond trees through different physiological stages or different seasons of the year. Before I begin, so this work is part of the work that's been done in the National Consortium of Xylella fastidiosa in Israel, which includes a lot of research groups and also collaborators from the Plant Protection and Inspection Services. So I would like to thank all of them for their help and collaboration throughout this important project. Okay, so as you may have heard, Xylella fastidiosa was detected in Israel in almond trees some years ago. And not long after it was detected, one of the things we've noticed is that when we revisit infected trees during spring, we don't see any visible symptoms of Xylella. If you take, for example, the two trees that you see here in this picture, so on the left side, that's a healthy tree, and on the right side is an infected tree. So you can probably notice that the infected tree has less foliage than the healthy tree. But if you look closer on the leaves themselves, the leaves of the infected tree looks perfectly healthy, and there is no sign of any symptoms of almond leaf scorch on these trees. It's only later on in the season where you start seeing the typical leaf scorch symptoms, as you can see here. And then even later on, in August or September, the whole canopy of the infected trees becomes golden or scorched, and the tree looks pretty bad, as you can see. Now, these trees will later go into dormancy. And in dormancy, of course, you can't see any symptoms. And then in the following year, the cycle repeats. So new leaves will emerge. They will look perfectly healthy and no signs of symptoms. And then again in summer, they will show the symptoms. So if we try to look at it as a cycle in almonds, so during dormancy, of course, we don't see any symptoms. When the tree blooms with, again, no symptoms. Early spring, we don't see any symptoms. And then in May, we start seeing early symptoms, and then in August and October, full-blown symptoms in the trees. So part of the year, the tree are asymptomatic, and then in the other part of the year, the tree are showing typical almond leaf scorch symptoms. And we were very interested to understand how the distribution of Xylella within the different tree organs aligns with the symptoms that we see in different seasons of the year or in different physiological stages. So this is basically the driving question of this research. Now, the results I'll be presenting are the results of part of the thesis of a master's student in the lab, Noah, and we're done in collaboration with Professor Danny Steinberg. So the approach we took here was we selected 10 Xylella infected trees. So in Israel, we have subspecies fastidiosa, sequence type 1, okay? And we selected 10 infected trees, and of the same variety. And these trees were sampled throughout 2 complete years in different times, in different seasons, as I will show you in a minute. And we also selected 3 uninfected trees to be used as negative controls. Now, as for the seasons or the time where the tree were sampled, so we basically sampled the trees 6 times a year during January, when trees are blooming, and then March, when we have new flush, April, where the fruit is green, July and August, when we harvest the trees, then October, towards dormancy, and December, where the trees are dormant. So these are the 6 times where the trees were sampled every year in those 2 years. And the tissues that were sampled are the following: roots, flower buds, flowers, leaves, green stems and woody stems, green fruit, and dry fruit. Of course, some of the tissues could only be sampled in certain seasons, like flowers can only be sampled, of course, during blooming. But the tissues that can be sampled throughout the year, like roots and stems, were collected throughout the year. And here's an example of how those tissues look like when we sample them. So in the bottom panel, you can see the tissues as it was collected from the tree. And then in the upper panel, above the dashed blue line, you can see the actual samples that was taken for DNA extraction. Okay, so DNA was extracted from each of those samples and then was used for qPCR based on the method of Harper et al. And you can see that in the woody stem or in the green stem, what we took for the DNA extraction is strictly the stem. I mean, no petioles, no leaves attached, just the stem themselves, and all the other samples. You can see how they were sampled and examined. Okay, so let's start with the results. So first, as I said, we use qPCR to try and assess the incidence of Xylella in those different tissues and different seasons. So initially, we could not detect Xylella in the roots in none of the trees that we've sampled and none of the sampling points. And this relates also to the question that was raised before by Maria as to whether we can find Xylella in the roots in almonds. So we were never able to detect it in the roots in those 2 years of sampling. I will refer to that a little bit later again. We also did not find it in flower or flower buds and not in fruit or in fruit peel. So the tissues that where we did find Xylella are basically 3 and include the leaf petioles, the green stem, and the woody stem. So all the data I'll be presenting from here on relates to these 3 tissues. So let's look on the incidence of Xylella in these tissues in different times of the year. So let's first look on leaf petioles. That's the orange line. So you can see that during February, the incidence is around 40% or 50%, which means that in some of the leaf petioles we were able to detect Xylella, but in some of them we were not able to detect it. And as we go into summer, the incidence of Xylella increases to about nearly 100%. And then when we enter in fall, the incidence starts declining a little bit in the leaves. Now, on contrary, when you look on the woody stem, that's the gray dots you see at the top, you can see that we were able to detect Xylella in the woody stems in all sampling times, and you can say in 100% of the samples that we have collected, which means that Xylella was present in those tissues throughout the year in all 10 trees. It's important here to mention that all the trees that we've selected for this survey were highly infected. When I say highly infected, I mean that more than 50% of at least 50% of the canopy was showing obvious disease symptoms. Okay, so these were trees that were apparently infected for quite some time. And with the green stem, we saw something quite similar to the woody stem. Okay, in most of the cases, we were able to detect 100% of the time Xylella in the green stem. Next thing, we also wanted to look about the relative concentration of Xylella in those different tissues and throughout the year. So we can see something that kind of complements the previous graph. What we have here on the y-axis is delta CT. So we use the normalizing gene from almond here to normalize the results. And the higher the delta CT value, the lower the Xylella population is. So again, if we start by looking at the leaf petiole, we see that initially in February, March, the concentration of Xylella in the leaves is very low. As I said before, in some of them it's undetected. And then as we go to summer, the concentration of Xylella increases quite significantly. And then entering fall, the concentration starts decreasing again. And as we saw previously for the incidence in the woody and green stems, you see that the concentration of Xylella, at least based, of course, on DNA and the qPCR, the concentration is very constant throughout the year. And it's quite high because if you look on the peak of Xylella concentration in the leaf, which occurs around July or August, it's not that far from what the concentration is in the stems throughout the year. Okay, now I also wanted to try and complement the qPCR method by trying isolation, try to extract bacteria from these different tissues in the different time points. So this is of the results of leaf petiole that were sampled in spring, summer, and fall. And as you can see in spring, we had 0 successful isolation from leaf petiole, and then in summer around 80%, and fall around 70%. So this also aligns well with the molecular incidence of Xylella in the leaf petiole tissue, and as we saw in the previous graphs, and we also see here. In the green stem, we saw quite constant success with isolation, around 50%, and in the wood stem, a slightly lower successful isolation, especially in spring and summer, as compared with the leaf petiole. Now, on top of isolation, Noah also tried to employ fluorescent in situ hybridization technique to identify Xylella in the leaves. And here too, what you can see, look on the first on leaf petiole, you can see that in summer, in 46 samples out of 47, we could see Xylella incidence in the tissue. In fall, it was much lower. And in the green stem, the incidence of Xylella was significantly lower than the petiole. And in the woody stem, we were not able to detect Xylella at all using this method. We'll talk about it a little bit later in the discussion of why that may be, but why the leaf petiole correlates quite well with what we saw with the qPCR in isolation. The green stem and woody stem, where we had 100% detection using qPCR, you see that in the fish analysis, it doesn't really correlate with that. Okay, if we try to summarize this, we've never detected Xylella, as I said, in the roots of the trees that we've tested, and that's a very interesting point. Actually, it was very funny to see Maria's question. She was mentioning the rootstock GF677, which is also the most common rootstock used in Israel in almonds. And one of the hypotheses we have is that maybe this rootstock serves as a barrier for Xylella, and it cannot migrate past it into the roots, because maybe this rootstock is resistant. We know that this rootstock is actually a hybrid between almond and peach. And peach is resistant to the ST1 strain that we have here. So maybe this hybrid in itself is resistant, and this is one possibility why we don't see it in roots. We currently are conducting experiments inoculating this the rootstock itself to see whether it can be infected and colonized by Xylella. And what we saw in the leaves, this is basically the main tissue where we see some kind of oscillation during the year. Okay, in early spring, the incidence of Xylella is quite low. It then increases gradually, and it peaks in summer, and then in fall, the concentration starts decreasing. And on the woody stem and green stem, on the other hand, the concentration is very constant and can be detected very reliably throughout the year, even during dormancy. And this method actually has been used by us if we need to check the trees in times where we can't see see symptoms. And despite this constant and relative high concentration of Xylella in these woody stems, it was difficult for us to isolate bacteria from there and also to visualize it by fish. This could be a result also of technical issues, because it's much harder, of course, to crush and extract bacteria from lignified and hard tissue like the stems, as compared with leaf petioles, which are very fresh and soft. But this could also be a result of the different state the bacteria is in within those different tissues. In the leaves, it might be more free swimming and more active, and this could be a reason why it's more easy to isolate it from there, while in the more lignified tissue like the stem, it could be more aggregated or sticky. And generally, as I said, what we see when we think about this cycle, it seems like the main.
I'm having trouble hearing you.
Sorry, Siri woke up from my from my iPhone. The main oscillation that we see in almond tree is in the leaf petiole, where we see the bacteria, the concentration starts growing from spring into summer and then declines in fall. But in the stems, we see quite a constant concentration of the bacteria throughout the year. And then one question is, for how long can this cycle persist of reinfection and then dormancy? The leaves fall off. Some of the bacteria, of course, lost. Some of them are maybe retrieved back into the stems, and then in the following year they re-inoculate, or not re-inoculate, they inoculate the new flush. And this cycle seems that can go on, and it's really a good question for how long can this go before the almond trees may die, or it may never die, but just sustain this kind of persistent infection. And that's it. Thank you very much for your attention.
Thank you very much, Ophir, for your presentation. And I assume we will have some questions coming then for the discussion after the next two talks. Now it's my pleasure to announce our next speaker, Yanqi Chen from the US Department of Agriculture, doing research on crop diseases, pests, and genetics. So she comes from Raleigh, California, and will speak about genome rearrangements in Xylella fastidiosa and an update on Xylella taiwanensis. We are looking forward to listening to your talk, Yanqi.
Okay. Can you hear me now?
Yes.
Okay. Oh, I need to have this on. See my slide?
As well, yes.
Okay. I think I can move on from here. I'm going to talk about this genome rearrangement in Xylella fastidiosa strains in US, and the research update on the Xylella taiwanensis. Here are the some example of the Xylella disease in US and literally in California. This is Pierce disease on the left on grapes, and is associated is caused by subspecies fastidiosa. And I give two examples of there are a lot of strains, and example of strains in M23 I'm going to talk about is from California, and ATCC 35A79, which is a type species specimen from Florida. So on the right is the almond leaf scorch disease.
Sorry, Yanqi, to interrupt you.
Yes.
Can you please change the display settings of your PowerPoint in the black window? You should go on display settings. You should see this button at the top of your PowerPoint. Yes. No, display settings, display settings. Yes. Click on it and then swap presenter view. Swap presenter.
That look good?
Okay, now it's fine. Thank you.
Sorry for that. I'm confused. that's example of Xylella disease in California. This is almond leaf scorch on the right, and it's caused by two subspecies, subspecies fastidiosa and subspecies multiplex. On this side. So when we, I've been working with the characterization of Xylella, and since my graduate work, as a matter of fact, a while ago. Usually, I remember the equation that a phenotype is the sum of genotype and environment. So in the past, we've been doing a lot of work on the phenotype, not because I want, but that's what we can do in those old times, that you do culture, you do description, whatever. Then the technology start to improve, and we are able to work on DNA, and particularly the sequencing technology improved so much, so we go to the genome. And also the other part is the environment. I call it environment, which is an easy thing to see, but it's kind of difficult to, well, we still don't know what to do with it. What's the best way to describe the environment associated with the growth of bacteria or the disease? But anyway, I'm going to focus a little bit more talk on the genome. But before that, I want to show a slide on the phenotypes research a while ago, and it's a phenotypic variation of Xylella. The phenotype is good; it's the most obvious thing to see, but we consider that as not stable because you may change, depends on how you set it up the condition. So here is a colony morphology of a multiplex, subspecies multiplex, and here the middle is a morphology of fastidiosa, and you can clearly see the difference. And in fact, this is very interesting. They call it the twitching motility or whatever. There are a lot of work have been done, but there are still a lot of work can be done if you want to do it. And interesting thing here is I mix the two subspecies and inoculate into a plant, and you can isolate that the two bacteria strains, and you can tell just by morphology in this under this condition. So now, since the phenotype we consider to be not that stable, and that's why one of the reasons to go into the genotype. So to work on the genome, and the current GenBank has 126 whole genome sequence of Xylella, and 31 of them are in the complete status, and there are 53 plasmids. So all this work is related to the sequencing technology, particularly the next generation sequencing technology. Now, in this next generation, it is NGS, and there are two types. I classified them. And one is the short reads based sequencing, which is 454, Illumina, Ion Torrent, whatsoever. And another more recent one is the long reads one, which is PacBio, Nanopore. And it is the use of the long reads sequencing, well, actually combined with the Illumina to get quite often, that make the whole genome sequence, the complete genome sequence of Xylella easy. So that's why we start to get a lot of so-called complete status sequence. With the genome sequence, we can look into the genomic variations, and I try to summarize in 4 types. And the one is the SNPs, single nucleotide polymorphism, indels, a short sequence, insertions, deletions, and CNV, copy number variations. SNP and indels has been commonly used, knowing it or not knowing it, like PCR parameter designs and whatsoever. But this type, the structural variations, has not been fully explored. But with the whole genome sequence available, and we can move forward to look into the variation at the whole genome structural level. I also call it genome rearrangement. That's why we did the work, and we actually published this work, the whole genome sequence of Xylella fastidiosa ATCC 35A79 type specimen, the type strain, and detection of the genome rearrangement within the subspecies fastidiosa last year. It's kind of interesting thing because of the whole genome is available, and when you compare the same strain, the M23 versus M23, and you do it in the dot matrix method, and you can see this diagonal thing. But that means it's linear, the same, nothing changed. The same thing when I did the A79 against A79, you see the same. Well, of course, they're comparing to themselves. Now, when you compare the M23 with A79, this is what you see. And they show me that the rearrangement occurred. There is genomic rearrangement or structural variations. So the way to interpret it is like that. From A79, that the order is A, B, C, D. Now you look at the M23, the order is A, C, B, D. Now if you look at every gene, they are almost identical. But if you look at the structure, well, the two things are not the same. So this is quite an interesting thing that we found, that also that we zoom into the junction area. You see a lot of phage-related genes in this area. The same thing here and here too. So that's saying with that they will assume that the rearrangement is mediated by phage or prophage activity. Now, this is the subspecies fastidiosa. That's what I'm talking about here in the publication in 2020. And what about multiplex in fastidiosa? And yes, I don't want to show a picture here, but it's in the publication. Because it's more or less the same type of variation, but different. Now, what about others? In fact, that the porca and the fastidiosa SVs has been reported a while ago in this publication, and they compared the CVC strain of citrus to PD strain in California. Now, what about the porca? Since there are a lot of whole genome sequence in GenBank, I look into it already. The answer is yes, but I don't publish it. I'm waiting for the colleagues in Europe to look into it and make a publication, or maybe they already published. I don't know. But anyway, it's a new thing. And what about the multiplex against multiplex? The answer is also yes, absolutely, as a matter of fact, because you look at the name multiplex, that means multiple things there. So there could be more subspecies you can go into, and there are already some proposals. The only thing is that for those proposals, you need to substantiate with a description, with an experiment, with a paper, not just bring up the name, but some more work need to be done. So a lot of exciting work is going on. Now I go down, since that I compare the fastidiosa against Taiwanensis. The Taiwanensis in GenBank, there are 4 genome sequences, whole genome sequences there. 2 are complete. So I compare this type strain, PLS 299 from the Taiwanensis against the 35A79 type strain. And yeah, you can see it's expected, as a matter of fact, but it also shows the power of the SV research using whole genome sequence. Well, with this, I'll just continue the update on the Taiwanensis, which is a species described by Su et al. In 2016. So this is pear leaf scorch disease in Taiwan, and see the scorching leaf, and I call it marginal necrosis. And this is the way they grow pear in Taiwan. So environment is important. This is kind of interesting. You don't go to Taiwan, you don't know that. You see a pear is a tree, you know, but they grow like a grapes with trellises. Interesting thing. Now, also, I went to Taiwan and worked with them, and we look out for the vectors and try to find out what is the vector. This is the issue here. In 2002, the college in Taiwan already get the PCR method on that. Of course, they publish in Chinese, but if you read it, it's not difficult to me. It's stuff from rapid analysis, use LPA primer, and get the sequence of 1,400 base pair, and sequence it, and design primers set, and use PCR and amplify specifically a 416 pp amplicon. So it's interesting to work with them. But for sure, there are more PCR detection techniques on the way. And one of the questions that a lot of you are concerned is, what about the natural vectors? Work has been done, but numerous insects, including 450 Cola palula with palula, the leafhoppers of Pierce disease, Xylella, as a matter of fact, were collected and PCR tested, no confirmed. Nothing is tested. And also it collect over 1,000 plant samples and did PCR, no Taiwanensis detected. But here's another update on the USDA and Taiwan collaboration. We work on the metagenome, which is in my lab mostly. And we got the DNA, did the NGS, and did the sequence analysis. There are quite a few discoveries, as a matter of fact, but this is one that I want to talk about, that is a small plasmid, around 2 kb plasmid detected, and 4 open reading frames, only 1 can be annotated. And this plasmid is not in the whole genome sequence that reported in GenBank. So when we do the phylogenetic analysis, and you see the Taiwanese plasmid is between Xylella and Xanthomonas, which is the same thing for whole genome sequence comparison and also the 16S rDNA analysis. This is interesting thing, and a lot of work need to be done, but it's still we just started it. So, in summary, that for Xylella taiwanensis, it's there and it's interesting, but still a lot of work need to be done to understand the biology of it. I think that's all. I want to thank you guys from California, and also want to thank you guys, help my colleagues from Taiwan to say hi, say thank you to you guys.
Thank you, Dr. Chen. We can move to another short talk of our Young Researcher Initiative. It's now the turn of Nicolas Pavlin, affiliated for Génie Enzymatique Cellulaire, CNRS, and Université de Technologie de Compiègne, France. Nicolas will talk about phage display as a new tool to tackle Xylella fastidiosa. Please, Nicolas, I leave you the floor.
Thank you. Thank you for the introduction. Can you see my presentation?
Yes, perfectly.
Okay. So thank you everyone for attending to the presentation of my research project, which is entitled A New Application of Phage Display, Development of Tools to Fight Against Xylella fastidiosa. So in this project, we focus our work on LESA, which is one of the most abundant protein of Xylella fastidiosa secretome. Researchers have shown that it is highly involved in Xylella virulence, as there is a correlation between LESA secretion and apparition of symptoms such as leaf burns and necrosis. So LESA is produced by the bacteria, then it is accumulated in the periplasmic space. where it can be internalized in outer membrane vesicles. And then in these OMVs can transport virulence factors out to the plant cell membranes. So LESA is a lipase with an esterase activity, and it comprises rigid 7-alpha helices lead domain. So as you can see here on the 3D model of LESA, which is a prediction model based on Xanthomona oryzae, one of its cell wall degrading enzyme. So in this model we colored the catalytic triad in pink here and the lead domain in purple. You may also see part of the single peptide here in red and some disulfide bonds in yellow. So this rigid lead domain forms a pocket in which the substrate will enter and dock. And at the aperture of the pocket, the active site of the enzyme is located and may cut the bound substrate. So this project is divided in 4 steps. First, we produced and purified LESA in E. coli model. And then we want to select peptides that are binding to our target protein using phage display experiments. Then we will study the peptide LESA interactions, specificity, and also the inhibiting power of these peptides related to LESA activity. So what is phage display? It's a biotechnological method used to study interactions between peptides and a target protein, or between antibodies and a target antigen. The technique is based on molecular expression at the surface of phages, creating large libraries of molecularly diverse peptides or antibodies. So in our library, all peptides are cyclic and of 9 amino acids in size, beginning with a cysteine and ending with another cysteine for cyclization purposes. And all these peptides are fused into the protein P3 of M13 phage, as you can see in red here. So selection of peptides interacting with our target LESA is done by biopanning. First, we immobilized LESA onto solid support, which is magnetic beads. And then we put it in contact with our library of phages. And the phages that will bind to our target protein will be kept, purified, and amplified, which enriches our pool of good peptide candidates. So the principle of biopanning is that each round of selection will improve the selectivity of the technique and also lowers the diversity of phages and peptides in our library. Selection pressure also increases with each round with the affinity of the peptides and phages with our targets. As you can see here, the figure which shows the library enrichment with each round. After few round of selection, we will sequence the phages, selected phages, to find out the selected peptide sequences. And then with these selected peptides, we will use them in various tests, in vitro and in planta, to study the selected peptides to LESA interaction specificity, and also to find out if these peptides could inhibit LESA activity. The end goal would be to find peptides that could help the plant to fight against Xylella infection and spread. And thank you very much for your attention.
Thank you very much for this introduction into bacteriophage display technology and application. We have now time for like 12 minutes for discussion, and we got a few questions from the auditorium. I have the first one going to OLeary, where it is asked, could this pattern of the infection, different infection over the seasons, be different for another subspecies like multiplex? Could it be different in regions with different weather parameters?
That's a good question. Of course, we could not examine that because we don't have multiplex in Israel. I guess as far as the distribution to the roots, that could determine on the compatibility of the specific strain and the rootstock that's being used. But of course, as I said, I cannot say anymore because we don't have multiplex here.
Thanks a lot.
Here, another question came from the audience. They're asking if, do you think that Xylella in Hula Valley doesn't spread by vector? And if you know if any study was done on vectors.
Yeah, so yes, there is a group of entomologists in our consortium that's been working exactly on that. We don't have any clear evidence so far that whether the disease spreads by vectors or not. I can say that we have detected Neophylenus campestris in Israel, a few individuals, not too many, but they didn't carry Xylella fastidiosa. And as I said, we don't have any proof at present that whether the disease is spreading with a vector or not. But our assumption is that it does.
Thank you.
I have a question to Yanji Chen. So it is asked, or it is remarked that you didn't find the Xylella taiwanensis in something like 1,000 samples. And the person is curious to know how many samples have been reported so far to be positive with this species of Xylella. Is this very rare or is this commonly observed?
It's well, on the 1,000 sample is the non-pair samples, because the pair there's no problem to get it, because they talk about the host range. That's why it actually it's been bothering us, because you keep on testing and then you don't see them. At the same time, they know that I didn't mention that there's also Pierce disease of grape in Taiwan. When you go to the grape, and for the subspecies fastidiosa, you can detect it in many plants and also many vectors.
Thank you very much.
I have one personal question to Nicolas. As you know, the catalytic triad, which is present in LS-A, is is widely common feature of lipase esterase families. So I guess if you have thought that potential control strategies targeting this domain could be somehow could have a wider range of potential targets also in other microorganisms or even in a plant itself. You are muted.
Sorry. I was beginning to explain that the cyclic peptides we want to find would be to maybe block the entrance of the lead domain to block the lipid or triglyceride docking in the lipase. As we don't know yet the natural ligand of the enzyme, we're not sure about what we are looking into the cyclic peptide sequences. But about the specificity of this enzyme is the lead domain. So usually the lipases have a few alpha helices which can rotate in order to activate or not the enzyme, meaning to allow the substrate to enter into the active site or not, depending on the hydrophobicity of the environment. And here, the fact that the lead domain is rigid and has 7 alpha helices and composed of 7 alpha helices is suggesting that the enzyme doesn't have any interfacial activation. And this is not specific to LESA, but also to lipases from Xanthomonas species.
Yeah. Okay.
So if you find cyclic peptide from LESA, we could use it also from lipases from other phytopathogens.
Yeah. Okay. Thank you.
I have a question to Ophir. It's which almond cultivars and rootstocks are the most affected in Israel? And do you observe yield differences between infected and healthy almond trees?
Okay, so most of the cultivars grown in Israel are local cultivars, so I don't know if you would know them, but all the ones that are grown in the infected orchards are, well, all of them, we have seen that all of them show disease symptoms. So none of the ones that we are using are resistant. As for the rootstocks, there are a couple of common rootstocks and there are some more or less common, but most of them, if not all, are hybrids of peach and almond. GF677, as I've mentioned, and Hansen is also a common rootstock, and there are some more. And as far as I know, they're all hybrids of peach and almond.
Mm-hmm.
Okay, there is a question to Dr. Chan. Which research laboratories are working, to your knowledge, on Xylella fastidiosa taiwanensis, in vitro or in vivo? You are muted.
Okay. Both Xylella fastidiosa, subspecies fastidiosa, and Xylella taiwanensis, they are in the central Taiwan. You know, they describe in central Taiwan. So one of the reasons is that there is a lab. It's one of the co-authors, Dr. Su. You know, you can check it. The lab there. But that lab is. They just apply plant pathology. Right? That's why they discover the disease and they come in contact with the lab in US and to do more research. So literally, that's the main lab. That's the main lab in working on Xylella taiwanensis. But they apply lab and they have interest to. That's why I get involved, and also University of Georgia, where I went to study and to work on the bacteriology level and also the genomic level. But I don't know others, as a matter of fact. There could be, I don't know. Yeah. Okay. So that's the situation because they apply lab. That's why you don't see a lot of publication, but they did a lot of work. So like they keep on testing, important thing. And that's why I receive email every now and then that asking me this question. Well, I said they just published, or they written it in Chinese, and you go to read the Chinese.
Okay.
Like
Thank you.
We have more questions for the phage library, phage display, and people wonder why did you use or choose a disulfide bonded constrained library and not a linear peptides? Is this a commercial library? What's about proprietary issues?
No, our library is not commercial. I wasn't involved in the creation of this library. It was inherited by another lab a long time ago. For the question about the cyclic peptides, so we are using cyclic peptide because it's a more stable form of a peptide. It cannot denaturate easily, and yeah, it's a mean for us to stabilize and also have the same cyclic peptide properties.
Thanks a lot. Massimiliano, maybe one more question?
Yeah, we have maybe the last question to Dr. Chen. Our attendees are asking what is the plant host origin of ATCC 35879 Florida? You are muted.
All right. It's great one in Florida. But it's sort of interesting that this is a type species. I assume all Xylella fastidiosa researchers should know it, but obviously it's not.
Okay.
Okay. Thank you very much to our speakers and people who asked questions for this round. And we are now coming to the last three presentations. Our first speaker will be Silke Robatzek from the Ludwig Maximilians University in Munich in Germany. And she will speak about a plant which is not considered typically a Xylella host plant. I think it's linked to Arabidopsis. It's your turn, Silke. We are curious to learn more about Arabidopsis.
Okay, thank you very much. I will start sharing my screen. Okay, so I hope it displays correctly. Thank you very much again, and I'm very excited to present our recent works on the interaction of Arabidopsis with Xylella fastidiosa. So our interest in my group is to understand the plant immune system and how it works. And basically, we can distinguish two different layers of the plant immune system. So the first layer is immune signaling mediated by PRRs, which are the pattern recognition receptors that localize at the cell surface. And these receptors recognize PAMs and DAMs for pathogen and damage associated molecular patterns. Then the second layer of the plant immune system is mediated by NLRs. These are cytosolic immune receptors, and they actually recognize effectors. And these effectors are typical virulence proteins which are injected into the plant cell from pathogens, and their main function is actually to suppress immunity, and the NLRs can detect actions. So the question that we were asking is, how does the immune system respond to Xylella fastidiosa? And our focus is the PRRs and their PAMs and DAMs. And the reason why we focus on the PRRs is the finding that Xylella fastidiosa lacks a type 3 secretion system and the type 3 secreted effectors. So this is typically the mechanism by which gram-negative pathogenic bacteria inject their virulence factors to suppress immunity, and Xylella fastidiosa doesn't have one. Secondly, if you consider the relatedness between a host and its pathogen, then we can assume that there are greater roles for the PRRs in more distant related, non-adapted host-pathogen interactions. And we hypothesized this is the case for Xylella fastidiosa, in particular in Europe, where the introduction was more recent and the host adaptation may be less specific. On the other hand, there are greater roles for the NLRs in more closely related adapted host pathogen interactions, which I said is mostly defined by the effector NLR interaction. So if we focus on the PRRs, there's quite a variety of different PRR structure types, which at the ectodomain enables the recognition of a quite large variety on their ligands, which can be the PAMs and the DAMs. And these ligands have different chemical natures. They can be proteinaceous, but they also can be, for example, oligosaccharides. So a very well-known PRR is the FLS2 immune receptor, which is the receptor for bacterial flagellin. And this receptor and the perception system is actually broadly conserved and present in a lot of plant species. So this is most plant species from the kingdom would actually recognize bacterial flagellum. But we know that Xylella fastidiosa doesn't have a flagellum. Another very well established and studied immune receptor is the EFR immune receptor. And this is a receptor that recognizes bacterial EFTU through immunogenic peptides, which are referred to as ELF18 or ELF26. Now, in this case, the EFR immune receptor is present in Arabidopsis and across the family of the Brassicaceae, but it is limited to the family of Brassicaceae. So in typical host plants, such as grapevine, host plants of Xylella, such as grapevine, this perception system actually is absent. So we decided nevertheless to use Arabidopsis thaliana as a system to study the interaction with Xylella because it is a well-established genetic model for plant immunity. So if we consider PRR immune signaling, they come in two different flavors. We can distinguish acute responses. And typical example is the generation of a reactive oxidant species as an oxidative burst, or the activation of MAP kinases, or also, for example, increase in pH as alkalinization. And another typical response is the chronic response, or better to say the response to chronic exposure to pumps and dumps, which is observed as the repression of seedling growth. So almost 20 years ago, me and my colleagues, we actually could show in this study in plant cell that the ELF26 peptide from subspecies fastidiosa is immunogenic and can induce this alkalinization response. And more recently, Alessandra De Souza and colleagues showed that the ELF26 peptide from subspecies Pauca is also immunogenic and can induce a ROS burst response, MAP kinase activation, and induction of defense genes. So what about a chronic immune response? So we have tested this, and we used the ELF26 peptide from the subspecies fastidiosa, and it can, when chronically applied to seedlings, repress seedling growth. So if these peptides, ELF26, which are derived from EFTU, are immunogenic, we were wondering how could they actually be exposed from the bacterium. And studies on OMVs from Xylella fastidiosa by Osias Paitosa Jr. and colleagues could show that actually the OMVs contain EFTU. So we could confirm this. We purified OMVs, and then we performed immunoblood analysis using EFTU antibodies, and indeed we can reveal a signal for the EFTU protein. Now, knowing that the ELF26 peptide is immunogenic and can repress seedling growth, we tested whether OMVs can do too. And this is the result that we obtained, is then when you expose seedlings chronically to purified OMVs from subspecies fastidiosa, they are able to suppress their growth. And now, taking advantage of the genetics in Arabidopsis, we could identify that this seedling growth repression is depending on the EFR immune receptor and its co-receptor BAC1. So this suggests actually that EFTU can be displayed by OMVs from Xylella fastidiosa, and its immunogenic component, ELF26, is being detected by the EFR BAC1 immune receptor complex, and that is necessary to induce the immune responses. Now, moving on to bacteria, we could also see that if we treat Arabidopsis plants with Xylella fastidiosa bacteria, this is inducing a ROS burst response, and this is greatly diminished in EFR mutants. So again, the ROS burst is also depending on EFR. And consistently with this, if we then monitor by PCR the abundance of Xylella fastidiosa in EFR and BAC1 mutants, we see a much higher abundance of the bacteria. And moreover, if we pre-immunize plants by pre-treating them with a ELF18 peptide again, and then subsequently, in fact, we can induce resistance of these plants. So the abundance of Xylella fastidiosa bacteria is greatly reduced. So collectively, these data suggest that the infection success of subspecies fastidiosa in Arabidopsis is depending on the EFR-BAC1 immune receptor complex. So then moving on, we were asking the question, how does Arabidopsis actually defend infection of Xylella fastidiosa? And to address this a bit more broadly, we used an untargeted metabolomics approach. So for this, we teamed up with the group of Corinna David at the Technical University of Munich and her co-worker Sebastian Bauer. And we performed several samples. So we used wild-type Columbia Arabidopsis plants and also the EFR mutants. And we either subjected untreated or mock-treated or fastidiosa infected samples at different time points from 2 and 8 dpi to 8 weeks. And then Sebastian actually generated 3 different types of extracts. One is ethyl acetate extract, then we have a methanol water extract and a water extract, and then subjected this to metabolome analysis. So in total, several thousand so-called features were identified after filtering, both in the minus and plus mode. And I think what is very nice to observe is if we cluster these features basically across the different samples, we can see that independent on the genotype, so whether it's wild type or mutant, and independent on the treatment time, which is either 2, 8 dpi or 8 weeks, we can observe that there's a clear clustering of features from the control mock-treated plants and the Xylella fastidiosa infected plants. So then it was necessary to molecularly identify these features to actually generate a database of Arabidopsis metabolites. So we screened all the literature which we could get our hands on, and we developed a database containing more than 3,000 metabolite entries. Now we use this database to then, as I said, try to identify some of these features for their molecular identities. And I should say that this database was generated by a student in the lab, Benedict Hempel. So I want to share some of these metabolites that we identified. One is phyophytin A and B. I should say this is preliminary because we're still awaiting our reference confirmation, but we are quite convinced that this is probably the case. This is why I'm sharing it here. And we are excited about this because increase, I should explain this a bit more. This you see an S-plot basically, and the dots below are the mock-treated samples, and the dots above on the S-plot are the basically is the increase in the infected samples, and the dots that actually are very high and to the right are very highly abundant in the infected sample. And for this reason, we were very excited about this because phyophytin A and B, when it's accumulating, they are products from chlorophyll breakdown. And this strongly suggests that actually subspecies fastidiosa, when infecting Arabidopsis, is inducing leaf chlorosis. And although we don't see this macroscopically, this is actually consistent with chloris observed, for example, in citrus and other host plants. So the second feature I would like to share is the maltotetraose and also maltotriose. They are highly abundant in the infected samples. And again, we are quite excited about this because maltotetraose is a product from starch breakdown, and its hydrolysis results in glucose, maltose, and maltotriose. So this could suggest that subspecies fastidiosa may enhance glycolysis in the plant, and we could speculate that in addition to glucose, maybe maltose may act as a carbon supply to support fastidiosa growth. And thirdly, I would like to share this feature, which is glucoraphanin. Again, is very highly upregulated in the infected sample. And glucoraphanin is a glucosinolide found in brassicacea. It is converted by myrocinases into sulforaphane, and the sulforaphanes have antibacterial activity. Now, Now, this is interesting again because it may indicate that subspecies fastidiosa increases glucosinolate breakdown, and maybe sulforaphanane may act against fastidiosa growth. So, excuse me. Overall, what we can see is that in Arabidopsis, Xylella infection actually largely restrict its development. So we see a macroscopic phenotype, which is this stunted growth and a much smaller inflorescence, and that's independent on whether it's a Columbia wild type or the EFR mutant. And this would be consistent with this increase a large metabolite reprogramming that, of course, it affects plant development. So the last question I would like to share with you is which other PRRs could be involved in immunity against Xylella fastidiosa? And for this, we are performing a candidate mutant screening. And this is prompted because when we monitored MAP kinase activation in response to Xylella bacteria, which you can see here is upregulated in a very typical pattern on 30 minutes and 60 minutes, this is not diminished in an EFR mutant background. So EFR is not required for Xylella-induced MAP kinase activation, suggesting that there are additional pumps and dumps that can be recognized. So our interest then turned to the WAC receptor kinases, which are the receptors for oligogalactonides, the OGs, which are plant cell wall-derived fragments and well-known dumps. And again, they occur in Arabidopsis, but also widely in other plant species, including grapevine and other host plants. Additionally, we were prompted because Caroline Roper many years ago has published that subspecies fastidiosa encodes several cell wall degrading enzymes, including a polygalactonase. This is required for its virulence, and polygalactonase degrades pectin and releases OGs. So then when we tested Arabidopsis WAC2 mutants, and you see two different genetic alleles here, the data are still preliminary, we need to repeat them, but nevertheless I wanted to share them with you, an increase in bacterial abundance based on PCR. So again, we are actually quite excited for this because it would suggest that also OG perception plays a role in restricting Xylella growth. So my summary, our data so far revealed that EF2 perception by EFR and BAC1 restricts Xylella infection success. Xylella infection results in metabolic reprogramming, and WAC2 could be involved additionally in immunity against Xylella. So our outlook is what can we learn actually, and can we use the knowledge that we have gained? And the question that we are pursuing is, can we engineer EFR expressing olives, and if so, could they be improved against Xylella infection? And this is prompted again by results from Alessandra de Souza, who could show that EFR expressing citrus shows improved immunity against subspecies Sauca. So with this, I would like to acknowledge the people who did the work. First of all, really outstanding and long-term postdoc in my lab, Kasia Rübak. She did all the pioneering work on establishing the Xylella Arabidopsis infection system, and also all the EFR mutant work and the candidate mutant screening. Secondly, I would like to acknowledge Benedict Temple, a student in the lab who developed the metabolome database. And I want to thank actually all of my group people, because it's a really inspiring, talented group, and I love working with them. It's great. Secondly, I would like to acknowledge again our collaborators, Corinna David and Sebastian Bauer from the Technical University for the metabolome works. And I would like to also acknowledge Jaffa from University Gießen, who is actually developing protocols for transforming olive. And I would also love to thank all the people from the community, and highlighting Stephen Lindo, Pasquale Saldarelli, Maria Saponari, Alessandra de Souza, everybody who helped us in providing materials and sharing knowledge. Thanks so much. It really, we appreciate this a lot. And lastly, I would like to thank our funding agencies, the German Research Foundation, DFG, and also the European Research Council, ERC. Thanks very much, and I'm happy to take questions. Thank you.
Our next and last speaker for the first session will be Wei Wei from the US Department of Agriculture, from the Molecular Plant Pathology Lab in Batesville, Maryland, and she will speak about the cold shock protein in Xylella. The floor is yours, Wei Wei.
Hello, my name is Wei, and I am a postdoc in Lindsay Burbank's lab at the USDA ARS in Carlier, California. There's actually another Wei in Beltsville, so that might be the confusion. But today I'll be talking to you very briefly about my project looking at a cold shock protein homolog in Xylella fastidiosa and how it's involved in stress response and biofilm formation. So our research station is located in the Central Valley of California. So we grow a lot of grapevines for wine and table grape production. So Pierce's disease, which is caused by Xylella fastidiosa, is a big problem for our region. So that's why we focus on the grape as our host. So Xylella, like many bacteria, encounter different environmental and host-derived stresses. So that can be abiotic or biotic stresses. And some mechanisms that bacteria have developed to counteract the stress is through the use of these cold shock domain proteins, which are basically nucleic acid binding chaperones that help facilitate transcription or translation during stress conditions. So traditionally they were induced by cold stress, but there are a lot of cold shock proteins that are temperature independent and actually induced by other types of stressors. And so they help destabilize secondary structures that may form during stress and then help facilitate translation and transcription. And some bacterial cold shock proteins have also been shown to influence gene expression. So in Xylella fastidiosa, there are two known temperature-independent cold shock protein homologs, so CSP1 and 2. And previous work done in our lab shows that if you delete the CSP1 gene from stag's leap, it reduced osmotic stress tolerance in vitro, as well as reduced bacteria titer in Chardonnay grapevines, suggesting to us that this protein is involved in stress response and or colonization or survival in the grape. And so we also know that CSP1 has general nucleic acid binding activity. And so based on our previous data and also data from other bacteria species, we hypothesized that CSP1 may be contributing to stress and survival by influencing expression of genes. And so to kind of answer that question, we further characterized the phenotypic differences between our CSP1 mutant and the wild type, and then also performed an RNA-seq analysis on these two strains using nanopore sequencing to identify if there were any differentially expressed genes and see what those genes might be doing in stag sleep. So then our first set of results, we noticed that there was an obvious difference in both cell-to-cell aggregation as well as attachment in the CSP1 mutant compared to our wild type and complemented strains. So as you can see in figure A, the images just show that the wild type and complemented strains have this ring of attached cells to the flask, versus the mutant is obviously deficient or has much less of that attached phenotype. And that is quantified in graph speed. And then instead of having this auto aggregation phenotype that the wild type has, excuse me, the mutant is dispersed and shows, more of a cloudy consistency in liquid culture, which is quantified in speed. And then we also were interested in seeing if the mutant was deficient in survival. And so we measured cell viability using a fluorescent viability stain. And we saw that at 7 days post-inoculation, there wasn't a significant difference between viability of all 3 strains. But at 13 days post-inoculation, which is well into stationary phase of growth, the mutant was significantly less viable compared to wild type and complemented strains. So suggesting that CSP1 is somehow related to long-term survival. And then moving on to our RNA-seq analysis. So we use nanopore sequencing to sequence transcriptomes of the wild type and the mutant under standard growth conditions. And we saw that there were 90 differentially expressed genes. And so one of the genes of interest that we saw was HILA1. It encodes a type 4 pillin protein and was upregulated in our mutant. And so previous work in other strains of Xylella show that HILA1 is involved in biofilm formation. And if you delete HILA1, it increased biofilm formation.
I'm sorry, you need to come to finish.
Okay, sorry. Okay, so yeah, we also noticed a similar phenotype. And so our conclusions were that CSP1 may be influencing biofilm formation, and this might influence plant colonization as well as stress. And then I just want to thank my lab and also the organizers and sponsors of this conference.
Thank you. We had indeed a very engaging talk. Several questions came from the audience, and I would start with a question to Professor Almeida. I was wondering how likely is the possibility that, according to your opinion, may occur to ST53 strain due to the large presence on the territory?
You mean what type of change may happen?
Yeah, if it's likely the possibility of further recombination events that may lead to some relevant phenotype.
Well, the short answer is, I mean, it's more likely than not recombining all the time, right? The only thing is that if you're recombining with the same, if you're exchanging a sequence but it's the same sequence, then you're essentially recombining with yourself, and you will never see genetic difference, right? So in the case of olive in Italy in particular, which is a young population, Ansi Carr gave a talk earlier, I think the first talk of the conference or something, and you don't really see a lot of recombination. And it's not that it's not happening; it's just that you don't see it because, again, you're exchanging genes with yourself, and the sequences are all the same, right? So I think that until you can gain or lose genes with other bacteria, that's a little bit different from homologous recombination. And you could, of course, if you have another introduction, but right now I think you're not going to see a lot of recombination events just because the sequences are all the same. Having said that, our work has shown that Xylella can change based on gene gain and loss, recombination and mutation. And much like COVID, you have such a large population size in Italy right now in particular, that I am pretty sure that you have strong selective pressures and it's adapting, as we speak. As to other phenotypes in terms of host plants, I think that it really depends on what's out there in the landscape, and I really think that large-scale monocultures do promote Xylella virulence, but you don't really have a whole lot of stuff other than olives, at least where I have visited. So at least for now, I think it may expand north, it may move, but yeah, that's kind of what I think. Thank you.
Okay, thanks.
Thank you. Michael Thieme has a question to Silke. So saying both glucosinolates and chlorophyll derivatives are known to play a role in herbivore defense. So do you think that induction of these metabolites could have an effect on the vectors?
Possible, but of course in our system we actually don't really work with the vectors. We do a needle artificial inoculation. And one should be reminded that actually glucosinolates are antifungal and also antibacterial compounds. So they're very important in defenses against microbes. For example, it has been shown that these sulforaphanates are actually they kind of reprogram Pseudomonas transcriptomes and therefore actually affect its virulence. So that could be also something to consider it in Xylella.
Thank you. There is another question for you. What is known about the presence of EFR and WOC receptors in the xylem tissues?
Yeah, so generally the receptors are expressed quite ubiquitously, and yes, we are interested in this. So Kasia has also generated EFR promoter gas lines to exactly look into this question, and from her first transgenic lines, the preliminary results would look like that they are actually expressed in the vasculature, and now we need to put it to a better microscope to see a better resolution of that.
Thanks.
And we got another question for you, Silke. In fact, the question is, does an EF26 Xylella knockout fails to induce an immune response? I assume it would be lethal to have a knockout of the gene, but do you observe sequence variation in the peptide moiety when you compare Xylella?
Yeah, so in fact, yes, an EF2 knockout is lethal. One could try to engineer an EF2 lacking this EF26 peptide because it's the most N-terminus. Whether this then generates a dysfunctional EF2 from its normal function, I don't know. So all what we know is that in prokaryotes, we have this N-terminal extension. In eukaryotes, we do not have it. So that's one interesting thing to consider. And this N-terminal extension is quite highly conserved. So there has been a study where they actually found signatures of diversity, which is basically trying to escape the recognition. But then, of course, you always have this trade-off that maybe then you generate a dysfunctional EF2 protein. So yes, indeed, this is something interesting, but the diversity will be limited. This is my my assumption on that.
Thank you. Thanks a lot.
I have a question also for Wei, and I was curious to know if you have any proof that your mutants can have also effects on pathogenicity traits in grapevine.
Yeah, so I didn't include that data, but yeah, so work done previously shows that the CSP1 mutant is also less virulent in the grapevine. So there is virulence data.
Okay, thanks.
I have a question to Wei and Silke, maybe both of you. It's about the culture protein, which is recognized in Solanaceous plants, like the culture protein from Pseudomonas syringae by the core receptor. What is known about an ortholog in trees like olive trees, almond trees? Do they exist? Can they recognize the culture protein? Maybe first.
I'm not sure.
Silke, do you know more about this culture protein detection?
Yeah, so in fact, we did some very basic sequence alignments to see whether the CSP22 peptide is conserved in Xylella, and it is the case. So one would assume that actually it is immunogenic. Whether it's core-dependent, meaning it's the Solanaceae type of receptor, or whether this is also, there's a related receptor in another species, which you cannot find by sequence similarity, we don't know, and that's definitely something interesting to follow up.
Thank you very much.
There's a very last question for Silke. Do you infer a possible glycolysis enhancement because of the highest availability of a substrate, as you said, provided by the starch degradation?
So this is data that we actually were thinking over the past recent months, so not so long ago, and we were quite struck by the finding that we have this very pronounced upregulation upon infection of maltotetraose and maltotriose. And reading a little bit the literature, it is interesting that maltotriose is a degradation, so maltotetraose is a degradation product from starch. And then maltotriose is generated from hydrolysis of maltotetraose. And of course, you should also then find back glucose in maltose, which may be something that just was failed to be detected. When I read the literature, it is interesting to see that actually phytoplasmas, which are also insect transmitted and colonize the phloem, both also induce basically upregulation of pheophytin A and B, so chlorophyll degradation, as well as upregulation on maltotetraose and maltotriose. And likewise as us, they were speculating that maybe maltose could be a carbon source for phytoplasma. So that's something interesting to think over.
Thank you.
Thanks a lot to all the speakers and all the participants in the discussion. I think we have to close this very interesting session. We have only 10 minutes left for a short break to have a coffee or a tea, whatever you wish. And we are looking forward to the next session, session number 2 of today.
Thanks, everybody. I'm sure that this session will give a relevant contribution to our next steps forward, a better understanding on biology and genetics of Xylella fastidiosa. It was really a pleasure. Thanks.